opportunity to tell you about some of the work that my groups and myself have done over the last few years. And I've really enjoyed the conference so far. I usually keep a running list of what have I learned during a conference, and I have about 10 different things I've learned during this conference. So I think that's probably a record. So I really approached this presentation thinking about what tools do we need to look at metabolomics data, and so what tools do we have currently in the tool set that we can use to look at the data and to use metabolomics to accomplish industrially important conclusions or results. So the structure of the talk, a little bit about POET. Probably no one in this room is familiar with POET, although you probably use gasoline in your car, and 10% of gasoline is ethanol, and POET is one of the largest producers of ethanol in the world. Talk a little bit about yeast biology. So almost everything that I work on today it comes from yeast. My wife says I'm going downhill. I started in pharmaceuticals working on animals and people, then I worked with chickens, and then I worked with C. elegans, and now I'm working with yeast. I'm not sure where I'm going from here, probably viruses or prions, and talk about some examples and the importance of experimental designs. We've heard that many times during this conference, and I'll reiterate the importance of the experimental designs. So POET's been in the biofuels industry, or the green technology industry, for 25 years. They currently produce about 1.6 billion gallons of ethanol per year. Our big project this year is moving away from starch-based fermentation towards cellulosic fermentation. Much more complicated process. They're a technology leader, and to a certain advantage, they have a single owner, a fellow named Jeff Broyne. So he started this on his father's farm, and basically he's grown. The advantage of having a privately owned company is that we're not at the whims of the stock market. So unlike the big companies I worked for, we don't have the ever-shrinking budget, and we don't have constraints that whiplash every quarter. So, people think that ethanol is new. Ethanol's not new. It's been around for a long time. The first cars ran on ethanol, and there used to be 2,000 service stations in the Midwest that sold ethanol. And gasoline is, it's a trend. We're mining petroleum out of the ground. It's not very green. It makes us sick. You just have to live in a city and deal with diesel fumes to know how bad petroleum can be. So the other problem with oil prices is we mine oil in places that that are far away, that require our military presence. It's relatively expensive. It's tough on the environment, and it impacts the standard of living. This slide is great because it shows the cost of a bushel of corn over the last 60 years compared to the cost of a bushel of oil. Of a barrel of oil. When you look at the oil companies, they make $50 billion a quarter in profit. That's incredible. That's more than the entire agricultural output of the U.S. When you look at how much does a soybean and corn cost. And we can produce a lot of ethanol from the corn. So this shows a typical biorefinery. Don't be misled by how simple it looks. This is very technologically sophisticated. In fermentation, there's 20,000 sensors that we have in that plant. We collect data on a sub-second basis, so it is big data. There is a lot of data. Not only do we produce ethanol, but we produce nutritionally enhanced feed at POET. So corn comes in. Corn is not very nutritionally complete. It's high in proline, which is an anti-nutritional. It has a lot of components in that evolved to stop the birds from eating the seeds. So we change the corn, we remove the starch, and we concentrate the proteins. But not only that, the yeast have a nutritional composition that is very similar to the needs of animals. So you end up with a much more nutritionally dense feed that the animals do very well on. We also produce corn oil fiber. When your Fiber One bars, probably the fiber came from POET or one of our competitors in the area, and other bioproducts. So a big one we're working on now is called Zine. It is a biopolymer that's biodegradable. Basically, it's not toxic, so it'd be great for paints and varnishes. And we're fortunate in that our owner really believes in science and technology, and he's a supporter. So I came to this conference because I've been working on metabolomics for a long time. I convinced my leadership that I needed to bring metabolomics to POET, and so we purchased a 6538 Agilent QTOF machine. It's a very nice machine, and we've spent the last 8 months developing methods and protocols to use this, and that was part of my goal of coming to this symposia. And we work with yeast cells, so the first thing I always do when I'm working on a project is I'm trying to understand the underlying organism. What is the systems biology of yeast? So there's a very nice diagram of what yeast looks like from an artist's conception. Often artist's conceptions are not very valid, so you want to compare it to what yeast really looks like. And so in the right you see you have the nucleus and vacuoles. When I think of a yeast cell, I think of a chemical manufacturing facility. So it's a biomanufacturing facility, and basically there's 21 unit operations in yeast that do different things like energetics and reproduction. Yeast has been used by man for thousands and thousands of years. I came across a paper in a science magazine that was talking about the Egyptians making a bread that contained yeast, enzymes, and starch, and you just mix it with water to make beer. And so that was 4,500 years old. It's amazing. So when you look at the biochemical unit operations, if you look at proteomics, this shows unit operations based on protein-protein interactions. There's about 21 unit operations. And a picture there, one of the things I'd point out in this picture is if you notice the nucleus, it doesn't have a very coherent nuclear wall. To me, that says it's old. I worked at Eli Lilly a long time on chronic disease, and basically as you age, you lose coherence, the ability to concentrate energy. It's all about concentrating energy. This is another view. This is from the cell cycle. This is from work out of the University of New Mexico, and it looks at the patterns of transcriptional profiling. Not surprising, there's 21 clusters here that you can pull out, again supporting that there's 21 unit operations in a yeast cell. If you look at humans, there's about 45, and plants are about 42 unit operations. And so, you know, the groups I've worked with, we've really looked to understand what each of the unit operations are, how can we modulate those unit operations, and how can we coerce the plants, or in this case the yeast, fungus, to do things that we want it to do, so to change how we go. This is a different view. This is actually, I stole this from Stuart Kim's work, but basically what you want to do is you like to understand the connections between these clusters, or the biochemical operations, the unit operations of the cell. If you understand what the connections are, you can get to the master regulators for the cell, which will allow you to change the metabolism. So at POET, if we can improve our yield by 1% by producing less yeast and more ethanol, it's worth $50 million a year, and it doesn't really cost very much to change metabolism. So there's a lot of money involved, and so there's a lot of incentive for us to be able to do it. Of course, as soon as the leadership sees that we've done something good, they really put the pressure on because they want more. So when I think of systems biology, I think of more of an integrated view, and basically looking at where we'd like to do. This is a slide from when I used to work at Eli Lilly, and we did systems biology in humans. But basically you have a knowledge base where you have what your a priori knowledge about the system, you have your in-life data, your compositional data, you have your metabolomics, and just like we've seen many times before, the metabolites will tend to cluster, and I'll talk more about the clustering in a couple minutes. You have your gene expression data, you have your markers, so basically where are the changes, the polymorphisms in the genes that make a difference. Almost always they're in the promoter regions or the introns, splicing regions. And you have your phenotypic assays. So when I think of working with high content data, it starts with a model. The only information you can get from an experiment is what you've designed in. So you have to design, you have to have the end in mind when you design the experiment. So if I have two treatments, A and B, I can separate in my metabolites or my genes into two buckets. Well, usually that's not too interesting. Usually we want to pull a lot more information from an experiment, particularly when they're expensive. By the time you fully processed a metabolomic sample, you probably have hundreds, if not thousands of dollars into it. So So you're talking expensive experiments. You want to make sure you get the maximum energy information content out. So one of the ways that I look at the data, this is a fermentation curve. So on the y-axis you have the ethanol titer. On the x-axis you have time. Turns out that this follows, can be modeled using a Gompertz model. Gompertz models have been around since the 1800s when the actuarials used them for lifespan estimates. And you can see that the experimental data points are the blue stars. The black dots are the predicted. It's not a perfect fit, but it's pretty good. And if you look at the model, let's see, I don't know if I can get a pointer here. So this is the genetic potential in terms of the rate. This is the genetic potential of the yeast to be able to deal with the stress of high osmotic, so high ethanol titers at the end. So you'll notice that we hit 20% ethanol titers in our fermentations, which is very stressful on the yeast. Of course, this was one fermentation. In the database I'm working with today, I have 57,000 fermentations at 500,000 gallon scale, so I have lots and lots of data to look at. Basically, the red line is the fitted Gompertz. The blue dots are the experimental data. You'll notice there's a little bit of a miss here at the bottom when we're adding the yeast to the fermenter, and there's a discontinuity at about 10 hours, which we're still really trying to understand. There's another one which is hard to see at about 40 hours. And it's shifts in the metabolism, and trying to understand those shifts is one of the things we're doing with metabolomics. So everybody, almost everyone that's given a talk has talked about principal component analysis. This is just a quick slide to talk about what does it mean, what does it look like. So basically, if I have a set of data, they often look like cigars. The first principal component is the length of the cigar. The second is the width. And if I have a three-dimensional data, think of a squash cigar. The third would be through it. Of course, we're talking about 21-dimensional data, right? If there's 21 unit operations in a yeast cell, that means there's really potentially 21 principal components. We don't often design experiments that can pick up all of them, but mostly we're interested in glycolysis. So if we have a poorly designed experiment, and I have some data here, so it has many features, in this case more than 2,000. There's not much information here. The noise will dominate the results, and it's pretty ugly. Whereas if I have a well-designed experiment, in this case I have time course data from a variety, about 25 different experimental treatments, then I can start to say something about the experiment. So the first principal component is related to metabolic rate. So basically we're modulating the metabolism. Actually, we add a little bit of acetic acid to our fermentation. What that does, that closes up the aquaporin channels, reduces the ability of the yeast to communicate with the outside environment, so it slows it down. And basically you can see, so that shows here, and then I've got a pretty extreme point up here. And then the second principal component is related to fermentation time. So if you have a well-designed experiment with clean data, most times there would be a very clear relationship between the principal components and the biological process. And so I have another example. This is 2,900 fermentations. And again, it's fermentation. You can see the fermentation as I move along the time, so my time course, it moves along this axis, and you can actually see, you can actually follow the principal components. You can also do this in human disease. So basically, if you look at the disease progression, you'll actually see those clusters move. How those clusters move is actually very important in understanding what's going on. And in this case, there are actually two different fermentation methods. So the top one is the POET no-cook fermentation. The bottom is conventional fermentation. So this was data from a plant where we had both types. And you can see there really was a shift in how the fermentation went and what happened to the metabolism. I'm only using a small number of metabolites here to make it easier to see and to understand. We have many more metabolites. So if we want to deal with many more metabolites, one of the methods that I've used is I call cluster correlation. It's really used to identify relationships in complex data sets. It's very closely related to principal component analysis, and it can be thought of placing the data into a standard canonical format, sorting a matrix to reveal the diagonal structure. It really works well if you have repeated measures data, and it's sensitive to the experimental design. And I thought we invented this in my group, and I found out the guys at NCI, John Weinstein and his team, actually had invented this before us, so you think you invent something, but often the case you haven't. So this shows some data from plants from a group when I was at Monsanto. And basically what you have is you have the metabolites on the X and the Y axis, and the data has been processed using the cluster correlation. And actually the metabolomics here is documented in a patent. The guys I used to work with went and started a company called Cantata, and they wrote a patent to actually get all the experimental details on how we generated the data from the Cantata patent that issued, I think, about 6 years ago. So I'm going to look at this little corner of this cluster correlation, and I'm going to show the TCA cycle. And what's important to note is, in this case, the red is positive correlation, blue is negative. But basically, molecular species that are adjacent in pathways tend to be very highly correlated when you look at time course data or when you look at data. So basically, in this case, we've picked up pyruvate, alanine, glutamate, alpha-ketoglutarate, oxalate. And so basically what we found is you can use this to really pick out the pathways. And if I do an experiment where I modulate the pathway, for example, we knocked out genes. So we did a lot of genetic engineering. We knocked out genes. These things light up like Christmas trees. So I can have two cluster correlations side by side, and I look at my treatment, a knockout, and they'll light up, and you can actually see. So we had, in one case, we had taken alpha-ketoglutarate, and we knocked the gene out and replaced it with something that had a much different rate constant for the enzyme, and you could see it. It lit up beautifully. So, of course, you'd think that you could take this data and construct pathways, and so I had a summer intern, and I asked him to look at pathways, and so we created synthetic pathways. We took that pathway data, and we generated synthetic data with and without noise. And what we found out is that the addition of as little as 1% noise pretty much destroyed the ability to recover the pathway. So unfortunately, you can't use this method to build pathways, but you can use it to compare what the data you're getting to your a priori knowledge about pathways. So a lot of work has gone into building the pathways, and this is a nice way to put the data on the pathways. In fact, you can take the nice, your metabolism map, and you can plot this data on it. I've used Spotfire as a pathway as a background to map the data, and that works really nicely. So talk a little bit about a different experiment. Again, I'm focusing on methods, so I'm bumping around between a lot of different experimental systems. This is a developmental curve for data. So I have one metabolite plotted over time in the top inset chart, and then transcriptional profiling data in the second chart. And if you generate a two-dimensional cluster between the metabolites and the genes, you end up with, again, a clustering approach. And of course, this is just 1% of the data, right? So there's 20,000 genes, 2,500 metabolites is what we saw in our discovery, of which 168 were directed metabolites that we looked at. And you can take these clusters, these blocks, and if you look at, you can generate the principal components and the loadings, and you end up with the same kind of clusters. I call these gene fingerprints, so mapping the genes and the metabolites together. You end up with a nice set of clusters, and this is just the first 3 principal components. There are about 9 that were significant in this work. And you can use a simple t-test to compare one cluster to another to see how it's doing. So the nice thing about cluster correlation is robust with respect to noise and signal strength levels. It's directly related to real pathways, although you can't recreate the pathways. And it's pretty straightforward. In R, it's like 6 lines of code to do it. So the challenge of high content data is you have millions of data points, lots of noise, missing data. So I'm going to basically, one of the ways you can look at the data, the challenge you have is if you have missing data, you're not going to be able to use a lot of the standard statistical methods because they assume there's no missing data. And in principal component analysis, if you have a piece of missing data, it drops the row. If you don't drop the row, you impute and put average values there instead. So missing data is really challenging. And so what we wanted to do is develop a different method to look at it, and it was we used what we call the percent change, or the distance from the line of no effect. Not sure. There we go. And that's the 45-degree diagonal. You can actually use trigonometry to calculate the distance. And these distances will follow a gamma distribution of order 2, or approximately 2. And you can actually use tools like Mathematica and SAS to calculate what the critical values are. And so now you can draw critical values. In many cases, the noise is a function of the signal strength. And so I have the shrinkage, which is the dotted line here. So it's robust with respect to missing data because I can calculate the distance in 2 dimensions, which is the example. I had 2 dimensions. In 3 dimensions, it's like a cigar-shaped region, and you can continue up. If I have a missing point, I can still calculate a distance, which allows me to deal with missing points, and then I use the values of the statistics in further analysis. So in respect to time, I'm going to skip the next section. So kind of noise has an impact on many of the tools. You really need to find tools that are robust with respect to noise, and you want to skip this one too. So you need to be able to have methods that will allow you to work without noise. You need to have an experimental design that has sufficient information content to allow you to be able to pull out a signal that's meaningful, and you'd like to understand the precision of the data. So what we found is that when you have noise in your data, you can introduce bias, and it reduces the amount of signal that you can see. You get a bias. I didn't have time to go through those slides. And it's also important to have funding for your research. And in our case, we have funding from POET, which provides forward thinking and the ability for us to really improve in the biofuels and the biomanufacturing area. So with that, I'll stop.
So I have one comment and a question. So I wish that you were at my class yesterday. You could have taught it. We had this project on biofuels, and so that's a senior design at Yale. So 24 hours late, but that's okay. And the other comment is actually 2. So why is there a lag in switching from switching to cellulosic feedstock? I mean, the data's out there that this is not a viable path. So is that just a lag in the technology at the plant, or what's the motivation for not getting to cellulosic feedstock like now?
So POET has a plant. It's called our Liberty Project at Emmitsburg. So we are bringing up unit operations even as I'm here.
Yeah.
So it's going to be either the first or the second full cellulosic plant. Currently, the enzyme cost is about 60 cents a gallon for cellulosic. Mm-hmm. And so over time, that would reduce. To produce a gallon of starch-based ethanol, the enzyme cost is about 4 cents a gallon. So we have to get the enzyme cost down. Also, we've done a lot of work to bring the feedstock to the plant. Right. So you need about 10 times as much stover or cellulose as you do starch. The energy density is much lower. So we actually are doing that. We are committed in the next 5 years to bring 5 plants online. So we work with a company called DSM to do that. So we are doing it. We're doing it both. We'll do it together with starch. So basically, we'll get the product from the plant. We'll turn that into high-quality feedstocks, and we'll take the leftover cellulose. We need to leave the nutrients and minerals behind and turn that into ethanol and other products as well.
And so, at least from what I gather from your talk, this overall analysis, although it's identifying important connections between nodes, but you're not able to leverage that to work toward design. So on the order of something like the work that Jay Kiesling's done, where he's actually increased flux to produce things like terpenes that look more like diesels. And so I'm trying to, I think for me there's a bit of a disconnect, and I can see and say Jay Kiesling's work where he's actually been successful in creating flux, but he's not doing it at the level that you guys are doing it. And I'm trying to figure out, so what's the lag in engineering these metabolic pathways?
Well, so part of it is, how we understand the biology today. So we have a number of projects where we're looking at engineered or designed organisms. I can't really tell you the company we're working with, but we're actually looking at producing other products. So, for example, butanediol is one that's widely used. We are also looking at basically at benzene ring type of compounds that are similar to biofuels. We do biodiesel, which is ethyl esters or methyl esters of fatty acids.
So yeah,
we think
you're
that
looking
is
at
actually
alkanes, fatty acid, yeah.
But today the problem with that is that the diesel motor manufacturers only allow you to put 5% biodiesel into the fuel before you'll invalidate the warranties. So it just takes time.
Sure.
One of the things I found working at Poet is I came in there, I said, I have all these great technologies that I learned in pharmaceutical business and the ag business. I want to apply them to the fermentation business or the biomanufacturing, and it just wasn't possible. It's slow, but we're getting there. I'm really pushing.
I understand.